What's up guys, good evening my chilla party. This is your NSEJS Biology Master Teacher Akshansh and welcome back to the next session for NSEJS 2025 Biology Basic to Advanced series. Students, we have already started with one of the most crucial and foundational units of NSEJS 2025 syllabus, that is cell biology. And this is the lecture number 4 for the same chapter. Well, in the very first part, in the lecture number 1, I had given you the introduction about the cell, the history behind the discovery of the cell theory and the factors that limit cell size or cell volume. In the lecture number 2, I had introduced you to the structure of the prokaryotic cell, a very detailed one where we had talked in depth about each and every single component of the prokaryotic cell. In the previous lecture, that is lecture number 3, I had taught you about the eukaryotic cell. We had defined what is a eukaryotic cell, the differences between the plant cell and the animal cell, and this is going to be in the sync with the same. Well, today we'll be talking about the other components of the eukaryotic cell. So let's start with the very first component, that is the plasma membrane. So what is plasma membrane? Plasma membrane is the membranous covering that is present outside the protoplast. Well, if I have to take you to the next level of scientific language, so plasma membrane can be defined as a quasi-fluid, pliable, elastic and film-like thin partition that is present outside and inside the cytoplasm. Now, very tough definition. Let me write it first for you, then I'll break down this definition into simple terms for you. Okay, so let's start with the very first definition that we taught you, that what is plasma membrane. So it is the membranous, membranous covering of the protoplast. Covering of the protoplast. Well, if talking about the characteristics of the plasma membrane, so the plasma membrane is going to be quasi-fluid. It is going to be quasi-fluid. It is elastic. It is pliable and film-like partition over and inside the cytoplasm, inside the cytoplasm. Okay, so in simple terms, what is plasma membrane? Plasma membrane, basically it is the membrane covering that is present outside the protoplast. And also, since we know that in eukaryotic cell system we have membrane bound cell organelles, so obviously the cell organelle, they are going to have a membrane outside them and the cell organelle, they will lie inside the cytoplasm. So that's why it is a film like partition over and inside the cytoplasm. So if I add all the membranes, right, the plasma membrane outside the cell and the membranes covering the cell organelles, so I can come up with a term that is the biomembranes. Okay, so altogether all the membranes that are in the cell system, they are called as biomembranes. So I'll write this term in caps, biomembranes. Correct? Now, so I have written some very fancy words over the screen, right? Quasi-fluid, elastic, pliable, film-like partition. So if you want to understand the meaning of these terms, we will have to look at the chemical composition of the plasma membrane first, right? And then I will tell you about the structure of the plasma membrane, and after that, all these terms will get crystal clear for you. Okay, so let's start with the chemical composition first. Right? So we are talking about the chemical composition. Well, the plasma membrane, it is majorly made up of lipids and proteins. In 1950s, chemical studies performed on the RBC membrane, that is the cell membrane of the red blood cell, it showed that the percentage of the protein is somewhere around 52% and of the lipid is somewhere around 40%. But this percentage varies from cell to cell. For RBC, it is 52% for the proteins and 40% for the lipids, right? So chemical composition, majorly it is made up of lipids plus proteins. Correct? Lipids. So primarily, let's take the example of RBCs. For RBCs, lipid percentage is 40% and for proteins it is 52%. So rest 8% that comprises of the carbohydrates and cholesterol. So remaining is carbohydrate. That is the 8% left. So that way it is going to be your carbohydrate, the sugar, carbohydrates plus cholesterol. Correct? Cholesterol. For the lipids, the primary one are the phospholipids. Okay, so the lipids, the primary one are the phospholipids. Phospholipids. And for the proteins, so the proteins that are there in the plasma membrane, in the biomembranes I should say, so they can be classified into two major categories: the intrinsic or integral protein, extrinsic or peripheral proteins. Okay, so the proteins that are there in the plasma membrane, they can primarily be classified into two categories. The first one is of the intrinsic protein and the extrinsic protein. Intrinsic or integral proteins. And extrinsic or peripheral proteins. Correct. So the intrinsic proteins, they constitute about 70% of the membrane proteins. So if I have to tell you, so 70% of the membrane proteins are integral and 30% of the proteins are extrinsic. So very important point about the extraction, from where the question can be created in for NSEJS, right? So if I want to study a particular type of intrinsic protein, so I will have to remove it from the cell, right? So how can I do it? So if we want to extract the intrinsic or integral protein, we will have to rupture the cell membrane. So you will have to completely disintegrate the cell membrane, and then only you can extract the integral protein. The intrinsic proteins, so the intrinsic proteins, they are actually embedded in the phospholipid bilayer of the plasma membrane, and the extrinsic proteins, they are present superficially. So when it comes to the extraction of the integral proteins, you will have to completely rupture the cell membrane, but for the extrinsic protein, they can be easily extracted, right? The extrinsic proteins, they are easy to extract, but for the extraction of the integral proteins, you will have to completely disintegrate the cell membrane. Very important point and very crucial for your NSEJS exam. Okay, so we are already done with the chemical composition. Now phospholipids. Let's take a few steps back and let's go to the chapter biomolecules. So there we had learned in depth about the structure of the phospholipids, right? Can I draw it for you? Should I draw it for you? Yes. And then we can talk about the structure of the plasma membrane, right? So phospholipids, right? If you remember, like a triglyceride. So let's draw the structure of the glycerol first. Okay, so this is what? This is your trihydroxypropane, that is glycerol. Okay, so glycerol. So you will have to establish two ester bonds with the carbon number 1 and the carbon number 2, right? Two ester bonds, so two esterifications with the fatty acids. Okay, so let's do it. R1, C double bond O, R2, correct? With the carbon number 3, so I'll remove this thing, I'll add a phosphate group, phospholipid. So I'll have to add a phosphate group over here. Correct? And here you are going to have the choline. We are making lecithin, which has the choline group attached to the phosphate group. So I'll remove this oxygen and I'll add choline over here. So here you have choline. So this is the structure of the phospholipid. Let's say lecithin. Choline will give you lecithin. Okay, so now if you look, let me just highlight a part of this. Okay, what can I do is I can remove this R1, R2. I'll put it here. Okay, I've just extrapolated the bond R1, R2. Correct? So the bond only. Okay, so let me just circle this part. Now, here you have the hydrophilic head, and R1, R2, these are going to be long hydrocarbon chains. So they are the hydrophobic part of the phospholipid, right? So R1, R2, these are long hydrocarbon chains, and this will make up your hydrophobic part. Hydrophobic tail, and here you have your hydrophilic head. Correct? A molecule having some hydrophilic groups and some hydrophobic groups, so such molecules are called to be amphipathic molecules, right? So the phospholipids, or simply the lipids, they are amphipathic molecules. So this is a amphipathic molecule, right? So now we are ready to discuss the fluid mosaic model that explains the structure of the plasma membrane, that was given by Singer and Nicholson in year 1972. Okay, so the next topic that we are going to be starting with is the fluid mosaic model. Fluid mosaic model. So this model was given by Singer and Nicholson. Let me just change the color of the pen. It was given by Singer and Nicholson in year 1972. So this model states that the phospholipids, they occur in the form of a viscous phospholipid bilayer. Okay, so I'll write this thing that they mentioned that the lipid molecules, first point of the fluid mosaic model is that the lipid molecules, they are present in a viscous, they are present in a viscous bilayer. And the protein molecules, so they occur at places embedded into the phospholipid bilayer. Okay, so proteins. What about the proteins? So they said that the protein molecules, they are going to occur at places now on both the sides of the phospholipid bilayer, right? On the outer side and the inner side of the phospholipid bilayer. So occur at places on the outer side and inside of the bilayer. So now how will it look like? It will look like the way the icebergs are there in the ocean, right? Icebergs that are there in the sea. Okay, so we can say the fluid mosaic model, if I have to call it in a very simple manner, I can say that protein icebergs in the sea of lipids, right? So if I have to define fluid mosaic model in a single statement, let me just take a good color. I think orange will work. Okay, so protein icebergs in the sea of lipids. Correct. So let's talk about the structure first. Yes, if you can look at over here, this is the detailed structure for the plasma membrane or for the biomembranes. If you can see this single entity, this is the lipid molecule. The circle, that is the hydrophilic head, and it has two hydrophobic tails. Okay, so this is the lipid bilayer. Okay, so this is one layer and this is the next layer. Okay, so now you need to understand that outside the cell you will have the ECM, extracellular matrix, and inside the cell you are going to have cytoplasm. Both ECM, extracellular matrix, and the cytoplasm, they have a very high percentage of water, right? Primarily they are made up of water. So water, when you have water, so the hydrophilic head, right? The hydrophilic head, it will always face outside towards the, for the outer side of the phospholipid bilayer, it will face towards the ECM, and the inner side of the bilayer, right? It will face towards the cytoplasm. The hydrophilic head, they will always face towards the water, and the hydrophobic tails, right? They will face toward each other. So that's how the phospholipid bilayer gets established. And if you can see, the proteins are there. The proteins are embedded, just the way the protein icebergs in the sea of the lipid. The icebergs in the ocean, the same way protein icebergs in the sea of the phospholipid. Okay, now if you can see, there are some proteins that are completely embedded in the plasma membrane. They are the integral proteins, right? I'll just highlight this. If you can see, look at here. So this is the integral protein. This is the integral protein. And if you can see, like some proteins present superficially, so they are the peripheral protein. So this is a peripheral protein. Now, very important, we had looked at some numbers, right? The chemical composition of the plasma membrane of the RBC. So they are 8%, right? 52% protein, 40% lipid, the remaining 8% that comprises of the carbs, the carbohydrate and the cholesterol molecule. So you can see, look at here, there you have the cholesterol. So this is the cholesterol molecule inserted into the lipid layer, right? And then you also have the carbohydrate groups of the glycoproteins. Okay, so the carbohydrates, they are attached to the proteins. So such a protein will be a glycoprotein. Okay, so the carbohydrate residues attached to the protein, glycoproteins. So this is the given structure is of the glycoprotein. Okay, so here you have the detailed structure of the plasma membrane. Very important, and you need to understand all these points very carefully. Okay, so we are done with this topic. Now the next, a very important concept, that is the asymmetry of the plasma membrane or of the biomembrane. Do you know that the membranes are asymmetric? Before telling you about the asymmetry, let's go back to the definition. So there is a term quasi-fluid, the quasi-fluid. So quasi-fluid term, it basically signifies the dynamic nature of the plasma membrane, right? The plasma membrane, the fluid mosaic model called the plasma membrane to be the protein icebergs in the sea of the lipid, right? So if you know that the protein icebergs, they can simply float over the ocean, right, over the water. The same way, they can move from one point to another. The same way, the quasi-fluid signifies the dynamic nature of the plasma membrane. That means the lateral movement of the protein is possible, right? The proteins can move. They can move laterally, right? If this protein wants, it can go to this side. Okay, either जा सकता है. It can go to in a particular direction. So the lateral movement of the proteins are allowed. That shows the quasi-fluid nature of the plasma membrane, right? Next, the elastic. So the plasma membrane is elastic, it is pliable, and it looks like a film under the electron microscope. So plasma membrane can be visualized only under the electron microscope. You will have to look it through electron microscope. So that's all about the plasma membrane. Now let's talk about the asymmetry of the plasma membrane. Okay, so we are talking about the asymmetry of the biomembranes. Do I have space? After this, we'll start with membrane transport. So I'll just write over here. What should I do? Okay, I'll just utilize this space. Correct. So we are talking about the asymmetry. Asymmetry of biomembranes. Do you know that the biomembranes, they are asymmetric? What does it mean? It means that the types of the phospholipid that are there on the outer side, they are going to be different than what are on the inner side. That means, let me just show you. So, okay, so this is the extracellular surface. This is the extracellular surface of the plasma membrane, right? Extracellular surface, and this is the intracellular. Let's see, intracellular. Okay, this is the intracellular, that it will be facing towards the cytoplasm, and the extracellular surface, it will be facing towards the ECM, extracellular matrix. Okay, so let me just color this one. So this is, I have colored it with green. So this shows that this phospholipid is actually lecithin. Okay, but if I look at the lipid molecule that is going to be on the inner side, it is going to be different, right? So basically, generally, the phospholipids that are there on the outer side of the phospholipid bilayer, they are lecithin. On the phospholipids on the inner side, they are cephalin. So lecithin, cephalin. Different types of phospholipids are present on the other two different sides of the plasma membrane. This shows that it is asymmetric. Rest. So I'll say, example, inner side में you have phospholipid cephalin. On the outer side, you have lecithin. Correct. So one example of asymmetry. The next Example of asymmetry, occurrence of the peripheral proteins. Peripheral proteins are generally more abundant on the inner side, right? Peripheral proteins, they are more abundant on inner side. Correct? More abundant on inner side. Madam, that means you will have more number of peripheral proteins on the inner surface compared to the outer surface, that is the extracellular surface. Next point about, if you can look at this diagram itself, you can tell that if you can observe these oligosaccharides, right, the carbohydrate residues occurring on the outer or the extracellular surface, but in the diagram there is no such carbohydrate residue present on the inner side of the phospholipid bilayer. Okay, that means the next is the oligosaccharides, they occur on outer surface, not on the inner surface. Oligosaccharide residues, you will never find any oligosaccharide residue on the inner surface of the phospholipid bilayer. Okay, so the 3 points that justify that the biomembranes are asymmetric. You will not find this much of depth in most of the books, right? If you want to get this much of depth about the concept, you will either have to refer to Campbell or some research papers, right? In general books, you will not find this much of depth. Okay, so I hope this information is going to help you a lot for your NSEJS exam. Let's move on to the next topic, that is the membrane transport. Okay, so obviously, if you know that the plasma membrane, it is selectively permeable. So the plasma membrane, we have been studying this since junior classes, that the plasma membrane is selectively permeable. Okay, so let's talk about the types of the membrane transport. Okay, so I'll just change the color of the pen. Types of membrane transport. Okay, so you can have three major ones: passive, active, and then you can have the bulk transport. Okay, so passive, active, and then you can have the bulk transport. For passive, you can have diffusion and facilitated diffusion. Passive shows there is going to be no expenditure of energy. Okay, so no ATP consumption. For active, ATP is required. Correct. For bulk transport, so it occurs in bulk. Okay, so for passive, it is diffusion and facilitated diffusion. Active transport, that is active transport, and bulk transport, so it is of further two types, that is your pinocytosis and phagocytosis. Pinocytosis, that is cell drinking, and phagocytosis, that is cell eating. Okay, so here you have the types of the membrane transport. So we can understand the types of membrane transport from this simple diagram. First, the passive transport, so there is going to be no utilization of ATP, no expenditure of energy. First is the diffusion. So for neutral or lipid soluble substances, they can easily diffuse out through the plasma membrane. Okay, there is going to be no involvement of any type of special proteins. Okay, so diffusion, that is going to be along their concentration gradient, so from high concentration to low concentration. Okay, so if you can see here, you have, let's say you have 5 dots. I'll just use the laser for this. Okay, so we are talking about diffusion. Sorry, no, I don't want this key, I want laser. Yes. So for laser, yes. So what is happening? Yes, so if you can see here, you have 5 dots, let's say 5 balls. Here you only have 2 balls. So high concentration to low concentration, if the molecule is either lipid soluble or neutral in nature. It can easily diffuse out through the plasma membrane. Now osmosis. So osmosis is a special type of diffusion. Again, the water solvent, it will move from its higher concentration to lower concentration is across a special channel known as aquaporins. Okay, so osmosis occurs through aquaporins. These are special channels through which the water can pass the plasma membrane. Okay, so that is that thing, that is osmosis. Now the facilitated diffusion. So what is facilitated diffusion? Again, facilitated diffusion is a type of passive transport. That means there is going to be no expenditure of energy, but for passive transport, some special type of channel proteins are involved. So because the channel proteins are getting involved, it is going to be highly specific in nature. So facilitated diffusion, it is highly specific. It occurs along the concentration gradient. So from high concentration to low concentration, the solute will move through their specific channel proteins. So the channel proteins, they are again of three major types, right? It can be either ligand gated, it can be voltage gated, or it can be mechanical gated. Well, this is, you will find this much of information in some very good research papers. Okay, so facilitated diffusion, it involves the it involves channel proteins. The channel proteins, so again I told you the channel proteins, so they can either be ligand gated, they can be voltage gated, or they can be mechanical gated. It will allow specific solute to move across the plasma membrane along its concentration gradient. Okay, so that is your facilitated diffusion. This is your facilitated diffusion. You can see the channel proteins are involved and the molecules, they are crossing along the concentration gradient. Then comes the active transport. Active transport, one very important example, sodium potassium pump, potassium hydrogen pump, right? So these pump, again, they involve some carrier proteins, they are generally called as pumps. Okay, so these carrier proteins are involved and they move, right? They move the ions, they allow the movement of the ions against the concentration gradient. Matlab, if a molecule, if a solute wants to go from its lower concentration towards the higher concentration against the concentration gradient, then it will have to go for active transport. In this, ATP is consumed. Okay, so this is against the concentration gradient. Okay, so the molecule is moving from low concentration towards the high concentration. So such movement is called as uphill movement. So this shows uphill movement. And for passive transport, high concentration to low concentration, this is downhill movement. So this is all about the membrane transport. And then bulk transport, so pinocytosis, phagocytosis, I would like to give you this as homework, as your research homework. So you have to research about, you have to read about pinocytosis and phagocytosis. I'll just drop you some terms that you have to look for in some good research papers, some good books. So pinocytosis, it is further of two types: selective pinocytosis and non-selective pinocytosis. Okay, so like you will find terms like receptor mediated pinocytosis and then phagocytosis. So you have to read about these terms. Phagocytosis is something that you have been studying for amoeba, right? Phagocytosis, cell eating. So pinocytosis, phagocytosis is your research homework. You have to research about these terms from some good research papers or from some good books. Okay, now let's move on to the next component, that is the cytoplasm. So we have already defined the term cytoplasm for the prokaryotes. The definition is still going to be the same. What is cytoplasm? So protoplasm minus nucleus, right? The protoplasm minus nucleoplasm is the cytoplasm. Cytoplasm, it is basically the crystallocolloidal complex that occurs inside the cell, right? It is the component of the protoplast. It is the component of the protoplasm and cytoplasm. So it is the crystallocolloidal complex. I'll write the same definition, right? It is a crystallocolloidal complex in water, right? So if you know that for cytoplasm 90% is water, so water is going to be the solvent. Okay, it is a crystallocolloidal solvent. Matlab, the chemical components that are going to be of the cytoplasm, they either they will make the true solution, right? Some chemicals, they will make the true solution with water, so you will get a true solution for them, and you will get colloid for some other chemicals, like say protein, RNA, vitamins. Okay, these substances, vitamins, amino acid, sugars, they make colloid solution, with water. Okay, so that's a crystallocolloidal. So the chemical, right, cytoplasm, everything is going to be dissolved in cytoplasm. So either the dissolution, right, you will obtain a mixture. So it can either be a true solution or it can be a colloid. So cytoplasm, it is the crystallocolloidal complex in water. It is the component of the protoplasm. It is the component of the protoplast. Simple. Correct. So that's all about cytoplasm. Now we can move towards the next topic, that is the endomembrane system. What is endomembrane system? So there are some cell organelles, some membrane bound cell organelles that function together, right? That function in a very coordinated manner. So this complete coordinated system is known as the endomembrane system. Okay, what I said, that some cell organelles, some membrane bound cell organelles work in a coordinated manner. Okay, so this system, this coordinated system will be called as the endomembrane system. So what are the major components of the endomembrane system? ER, Golgi body, lysosomes, vacuoles. So endoplasmic reticulum, Golgi body, lysosomes, vacuole, they make up the endomembrane system. Okay, so now if you can look at over here, so endoplasmic reticulum, Golgi body, lysosome, vacuole, I'll teach you how their activity are in a coordination, how they work together. So this makes up your endomembrane system. So we'll start with the very first component, that is your endoplasmic reticulum. Very first component of the endomembrane system, that is endoplasmic reticulum. So the endoplasmic reticulum, it was independently discovered by two scientists in year 1945, and the name of the scientist was Porter and Thomson. So endoplasmic reticulum discovered independently by Porter 1945 and Thomson again in year 1945. The term endoplasmic reticulum was given by Porter in year 1953. So term given by Porter in year 1953. Correct. Now what is endoplasmic reticulum? So it is a very complicated, three-dimensional interconnected system of membrane lined channels that run across the cytoplasm. Okay, so if I have to write the definition, what is endoplasmic reticulum? So again, it is a complicated three-dimensional interconnected network of membrane lined channels that run across the cytoplasm. Correct. So basically you have, let's say, a city. So a city has the sewer line, right? You have the whole complete sewage system. You have pipelines running across the whole city. The pipelines are interconnected, right? The same way. What is endoplasmic reticulum? These are the pipelines. These are the channels. These are membrane lined channels. Okay, so they are membrane lined channels that run across the cytoplasm. And the channels, they are very complicated. They are interconnected. They make a 3D network. Easy peasy. So you can look at the diagram itself. If you can see, you have channels, and these channels are making a very complicated, interconnected, three-dimensional network, and these channels are membrane lined and they are running across through the cytoplasm. So that is the definition of the endoplasmic reticulum. So the endoplasmic reticulum, as I said, these are channels. Okay, these are the pipelines. So the pipeline, let's say if this is a pipeline, this is a pipe. Let me just make a pipe. So this is a pipe, let's say. Okay, so you can see you will have something right outside the pipe and you have space inside the pipe. So inside the pipe, outside the pipe, two terms, right? Two aspects. The same way, the endoplasmic reticulum, it divides the intracellular space into luminal space and extraluminal space. Okay, so ER, I'll write this thing. ER divides intracellular space into luminal space, that is inside the channels, right, inside the ER, luminal space, the space that is inside the pipeline, and extraluminal space, correct? Okay, now extraluminal space. So ER divides intracellular space into luminal space, that is inside the ER, and the extraluminal space. So the two environments, right, they are going to have different composition, right? Chemically, they are going to be variations in the chemical composition, like the internal environment and external environment. Chemically, they about the activity, there are going to be certain differences. Now the next, the ER, if you look at the structure of the cell, if you can see it over here, okay, the ER, it at places it is connected to the nuclear membrane and to the plasmalemma, and it also passes through the plasmodesmata in case of plant cells as desmotubules. Okay, so it is attached to nuclear membrane and to the plasmalemma at some points, and it also occurs in the form of desmotubules across the plasmodesmata. This is what we have already learned. Okay, so should I write this point? Okay, let me just write this point for you. Okay, so it is connected with plasmalemma, that is a plasma membrane, and nuclear envelope. The next is, it also occurs in the form of desmotubules through plasmodesmata. Occurs as desmotubules through plasmodesmata. Correct? Easy to understand. I hope it is clear till here. Everything is clear till here. Now we can talk about the occurrence of endoplasmic reticulum. Okay. So we are talking about the occurrence, where and in how much quantity you can find the ER. So ER is very extensive in metabolically active cells, right? You will have a very extensive network of endoplasmic reticulum in metabolically active cells. So it is extensive, right? It is very extensive in metabolically active cell. Metabolically active cells, such as hepatocytes, hepatic cells, liver cells, pancreatic cells, etc. Right? It is simple in storage cells, right? It is simple in storage cells. So any example of storage cell? Adipocytes, right? Adipocytes. The cells making up the adipose tissue, right? It occurs in a reduced form, in the form of vesicles in spermatocyte, reduced in spermatocytes, right? Like it occurs in the form of only a few vesicles, right? It is very reduced in case of spermatocyte, and it is completely absent in case of RBC, absent in embryonic cells, it is absent in mature ovum. Okay, so absent in, and obviously prokaryotic cells will also not have endoplasmic reticulum. So absent in eggs, mature RBC, embryonic cells, prokaryotic cells, etc. Right? So very important, and you can definitely expect a question from this, a very good question, and many students will not be aware of this concept. Okay, so very crucial concept for endoplasmic reticulum, right? This data is very important because this much of depth is not given in general books, right? If you refer to class 9th NCERT, okay, 11th NCERT, or the books for ICSE board, or like the books just like the general books, right? The general sort of books. You will not find this much of data there. Okay. So now the occurrence, we are also done with occurrence. Then comes the types of endoplasmic reticulum. Okay. So next is, and here also we will talk about the function. So let me just change the color. Types of ER. Correct. So types of ER, two types, RER or SER. Rough endoplasmic reticulum, smooth endoplasmic reticulum. Do you know why rough endoplasmic reticulum and smooth endoplasmic reticulum? Rough endoplasmic reticulum, it is going to appear rough on its surface because of the presence of the ribosomes. So here you have ribosomes. Okay, but for ER, for smooth endoplasmic reticulum, ribosomes will be absent. Correct? Ribosomes absent. Now, what is the function of ribosomes? So they help to make protein. They are the protein factories. Ribosomes are protein factories, right? Ribosomes. So they are protein factories. Now, since over the SER, smooth endoplasmic reticulum, ribosome is absent, so it is also called as agranular endoplasmic reticulum. Also known as, so smooth ER is also known as agranular endoplasmic reticulum. Correct? It is also known as endoplasmic reticulum. Agranular endoplasmic reticulum. Now the smooth ER, what is the function? So it is involved in the formation of glycogen, fats and sterols. Okay, so what is the function of SER? SER is involved in the formation of glycogen, fats, sterols. Okay, so formation and storage. Formation, that is the synthesis and storage of glycogen, fat, sterol. Okay, I'll just write this thing as well. Synthesis and storage. Correct. Storage. It also helps in detoxification. It has certain enzymes that are involved in the process of detoxification. Now, talking about RER, rough endoplasmic reticulum, ribosomes, they are protein factories. So, primarily involved in protein synthesis. Correct. Now, so RER, what is there that helps ribosome to attach to the surface of the ER, right? But there should be something that is attaching ribosome to the surface of the endoplasmic reticulum, that is giving it the rough appearance, right? So what helps the ribosome to attach to the surface of the ER? Anyone knows? So certain glycoproteins are involved. They are known as riboforins. So riboforin 1 and riboforin 2, these are the special types of glycoproteins that help ribosome to attach to the surface of the ER. Okay, so I'll just write this thing, right? RER contains two types of glycoproteins. They are known as riboforin 1 and riboforin 2. Riboforin 1 and 2. Okay, riboforin 1 and 2. So that helps the ribosome to attach to the surface of the ER. Okay, so we are done with this thing, RER and SER. If you want to know about the structure, so it's very easy. Endoplasmic reticulum is primarily occurs in the form of vesicles and tubules, whereas the rough endoplasmic reticulum, it occurs primarily in the form of cisterna. Okay, so I'll just change the color of the pen and write this thing. A smooth endoplasmic reticulum, it occurs in the form of tubules and vesicles, but RER, it mainly or primarily it occurs in the form of cisterna. Correct? So here are all the details about endoplasmic reticulum. Now we can take a quick summary of what we have learned in the whole class. Okay, so first of all, I taught you the definition of what is plasma membrane. It is a quasi-fluid, elastic, pliable and film-like partition over and inside the cytoplasm. We use a term biomembranes to represent the membranes that are inside the cell and outside the cell, right? Then the chemical composition, we took the example of RBC. So basically the membranes, primarily they are made up of lipid and protein. For RBC, 40% lipid, 52% protein, rest 8% carbohydrates and cholesterol. Okay, so proteins, intrinsic or integral protein, extrinsic or peripheral proteins. Okay, Then we talked about the structure of the phospholipid, right? It is an amphipathic molecule, hydrophilic head, hydrophobic tail, that makes up the phospholipid bilayer. Okay, so the bilayer, and then there you will have the proteins, either embedded, integral proteins, or present superficially over the surface, extrinsic or peripheral proteins. Correct. Then we talked about the asymmetry of the biomembrane, right? So inner side, outer side, different types of phospholipid. Peripheral proteins occur more on the inner side. Oligosaccharides occur on the outer side of the phospholipid bilayer, right? Fluid mosaic model, protein icebergs in the sea of lipids, right? So that was given by Singer and Nicholson in year 1972. Then we talked about the membrane transport. Plasmamembrane is highly sensitive of the endomembrane system, ER, Golgi, lysosomes, and vacuoles, right? And then we talked about the very first component of the endomembrane system, that is endoplasmic reticulum, discovered by Porter and Thompson in year 1945. Independently they worked. Okay, the term was given by Porter in year 1953. What is ER? Complicated 3D interconnected network of membrane line channels that run across the cytoplasm. Okay, and then we talked about that the ER divides intracellular space into luminal space and extraluminal space. Connected with plasma membrane and nuclear envelope, it occurs as in the form of desmotubules through the plasmodesmata. So the occurrence: extensive in metabolically active cell, simple in storage cell, reduced in spermatocytes, and absent in eggs, mature RBC, embryonic cells, prokaryotic cells. Type RER and SER. RER, ribosomes will be there on the surface of the ER. So protein factories, ribosomes are protein factories. So RER is primarily involved in the process of protein synthesis. So basically there are some glycoproteins involved named as riboforin 1 and riboforin 2 that help the ribosome attach to the surface of the RER, right? For SER, so it is also known as agranular endoplasmic reticulum. Ribosomes are absent on the surface, involved in the synthesis and storage of glycogen, fats, or sterols. It also helps in detoxification. Okay, so SER occurs in the form of tubules and vesicles, whereas the RER, it occurs in the form of cisterna. Okay, that's all from my side for today's class. I hope you enjoyed the session. Thank you very much. We'll see you in the next class.